Hello and welcome back. We're going to continue on the topic of bacterial genetics, and the short unit right now is on transposons, discovered by Barbara McClintock, also known as jumping genes. And what they are is they're a piece of DNA that can move itself from one location to another within the same genome. They never exist independently. They're dependent upon being part of an existing genome, and in some cases they can bring multiple genes for resistance together into a single plasmid. So we'll talk about composite transposons, and what these are is they will bring resistant genes together and create new mutations, new combinations that weren't seen before. And as we just said, these transposons can increase the mutation rate, and they all require an enzyme called transposase. And the first thing we're going to look at is just the basic transposon, and what it does is basically it just copies itself over and over, and it's called an insertion sequence. And basically what it is it is the gene for transposase. And so an insertion sequence is just the DNA necessary for transposition, and it can create mutations. It can insert itself in the middle of existing genes. It can and cause them not to be expressed. And as if we see this diagram here, here's the target site, and we have transposons at the initial site, and what transposase will do will cut the DNA at that target site, insert a copy of the DNA in there, and now we have the transposon at a new site, and that can cause a mutation because it can interrupt the original target site. So here's an example of that, as we can see in this cartoon. Here's the transposon in purple, and what will happen is the transposon enzyme transposase will create this hairpin turn, and it will fold the DNA over, and it will insert itself into this other gene, that in this case green, and we get the transposon that is now jumped from one position to another and has disrupted this existing gene, and it may cause it to be inactive or not expressed at all. The other kind of transposon is a composite transposon, and here we've got the insertion sequence, but we also have genes for other traits, and here's where usually those other traits are going to be antibiotic resistance. So again, whether it's ampicillin, methicillin, we've heard about MRSA, but what happens here is when that insertion sequence, when it copies itself, it also copies a gene for resistance. And so this is one way that resistance to antibiotics can be transferred between different bacteria. And there's basically two methods of the transposition. The first is a cut and paste, number one here on the top, and in this case the transposon basically cuts itself into a new target site, pastes, and it has jumped, it has moved to a new area on the DNA. The other is a copy and paste mechanism. So here the transposon copies itself into the target site and leaves the original copy behind. So now it has basically replicated itself, and we now have two copies of that transposon in the same genome. And again, as we wrap up, the main thing here is the transposons can increase the mutation rate. It can give us new combinations of bacteria that we haven't seen before, and lets them evolve at a much faster rate than they may have otherwise. Thanks again.